Hi everyone, I'm Tim van de Bossche, and together with Professor Leiwan Lee, this great team of Metaproteomics researchers, and on behalf of the Metaproteomics initiative, we wrote the Microbiologist Guide to Metaproteomics that is now being published in iMeta. Let me first tell you something about the what and why of Metaproteomics. Metaproteomics studies a full protein complement of microbial communities, and it allows you to identify proteins and quantify their abundance in those communities. It also allows to link the identified peptides and inferred proteins back to taxonomy and function, and depending on your workflow and depth, allows you to detect post-translation modifications or PTMs and unravel its functions. Why is it important you incorporate metaproteomics in your workflow? Well, with metaproteomics, you can capture the active actual functions of microbial communities and are therefore ideally suited to complement other omics fields. It is therefore not a surprise that it has a wide range of applications, including microbial ecology, host microbiome research, improving biotechnological processes such as wastewater treatment, and environmental monitoring. Metaproteomics is even being applied to unique sample types, such as ancient microbiomes. In this comprehensive review, we cover all steps from experimental design to bioinformatics. After some introduction, we begin with providing you an overview on common metaproteomic experimental designs, followed by sections on sample collection, preservation, and pre-processing. This table, for example, summarizes commonly used protein sample preparation techniques with their advantages and potential disadvantages. Next, we discuss important steps in the wet lab workflow, including microbial cell enrichment, cell lysis, protein extraction, and peptide separation. Sample preparation can be performed manually or automated, each discussed in a separated section. Then mass spectrometry is used for data acquisition, and the resulting data are analyzed through bioinformatic pipelines. In the bioinformatics section, we'll go over some important concepts. First, we'll look at how we build databases, like you can see in this table. Then, we'll talk about how we calculate the false discovery rate using the target decoy approach. We'll also go through how we infer proteins from peptides and how we group those proteins. And next, we can see how we can infer taxonomy, either from peptides using the lowest common ancestor approach, like you see here, or from proteins. And finally, we'll wrap up with an overview of downstream statistics we can use. This is, of course, just a short version of the full outline. So I warmly invite you to read our review and, more importantly, to consider adding metaproteomics to your meta-omics toolbox. Before I finish this presentation, I want to spend some time to the Metaproteomics Initiative, an international community by and for metaproteomics researchers. The Metaproteomics Initiative currently gathers over 350 metaproteomics researchers from 95 labs in 23 countries and is largely based on 3 pillars. The first pillar is to increase communication and collaboration among metaproteomics researchers. We do that by, for example, organizing large benchmark studies, or commonly known as the CAMPI studies, and we help organizing the International Metaproteomics Symposium. On this picture here on the right, for example, that was in Avignon, France in April 2023. Secondly, we aim to improve education of metaproteomics for microbiome and proteomics researchers. This paper is an excellent example and will therefore serve as one of the flagship papers of our community. Next to this paper, we also regularly organize or help organizing workshops at major conferences. And last but not least, we are working on standardization. Most recently, we are working on the development of metadata standards in the field in collaboration with several other organizations. Now, to be able to do all these things, we are very grateful to receive support from the European Proteomics Association, EUPA, and the Human Proteome Organization, HUPO. In addition, we recently also established a collaboration with Elixir. If you want to stay updated about the community, I can only recommend you to become a member of the Metaproteomics Initiative. You can do that by scanning this QR code or go to our webpage metaproteomics.org.